Thank you so much for being here. Thank you to PacBio for introducing me or inviting me to give this talk. Jonas promised that I would be able to crowd surf off of this, so I fully expect the front row to catch me when I'm done. So today I'm going to present our some data of us and our journey with this fly, where we helped it reach its full potential of becoming a lot of different library types. So I'm Sheina. I'm a research biologist with the USDA Agricultural Research Service. Our mission is to create scientific solutions to agricultural problems, and we represent the research arm of the USDA. Our lab in Hawaii has been around since about the 1910s, and the reason for our existence is because of fruit flies. So fruit flies are species that are all over the world. They're found all over the world. They're specialty crop pests, and they're established in Hawaii, where we live. But they've mainly been kept out of the United States, so they're not established in the United States, and this is really important. One, because the specialty crop industry would really like to keep fruit flies from laying eggs in their fruit, and then so they can't sell it. So that's good that they're not there, so they don't do that. But also, them not being established in the mainland means that they maintain access to markets that are fruit fly naive. So we want to keep the United States fruit fly free. That is where we come in, so we do research to make sure that happens. Over the last few years, the USDA has invested very heavily on generating sequencing capacity. So from this, started two different research initiatives, the Ag100Pest and the Beenome100. They contribute to larger research initiatives where we sequence, like the I5K and the Earth Biogenome Project, where we sequence a lot of insects, both pests and beneficials, to generate genomic resources. ARS is separated into 5 different areas, and so our lab has a Revio, so we get insects from all over the country from these different research labs that we sequence and assemble. So this is a small subset of the insects that we sequence. We sequence way more than these. We sequence this, like I said, on the Revio, where we can do 4 plant or insect-sized genomes a day, and we like loads of coverage, so we budget about $1,000 per genome. To go through, to do something hundreds of times, I developed a genome assembly pipeline where we take HiFi data and then combine it with Hi-C data, end up with a really good scaffolded assembly. If you go through that entire pipeline, you end up with a genome that you could do lots of different fun analyses with, right? So you can look at your centromeres, your telomeres, you can do whole genome alignments, you can do synteny, you can look at orthologous genes and see what genes different taxa have in common, what genes are different, what are unique, and then using gene ontology, you can start to look at the function of those genes. But what if you could actually get more information from your one sequencing effort? So last year, my colleague Scott Geib came to this workshop and came to this meeting and presented the concept of a holistic genome. So where you take one insect and then from that get lots of different library types to get not just the contig assembly, but looking at it at a chromosome scale. You can look at the epigenetic regulation and also gene annotations. So if this is our insect, this is our fly, it has a head, a thorax, and abdomen. It was reared and then made into libraries by Angela and Renee, who are the best research biologist technicians that I've ever known. They're the most dedicated and the most talented, and if it weren't for them, none of this data would have existed as of like December. So they made all of this in a couple of weeks, and I analyzed it so that we could present it here and there. They're quite amazing. So they took this fly and then separated it into these different tissue types, ground it into a fine powder, then split that sample where half of it got treated with tri-reagent. So these tissue became a Fiber-seq library. So as Jake mentioned, you do like a methyltransferase treatment on the nuclei. This tags the A bases that in open chromatin, so that way you can identify what is open once you do your assembly. And then it gets sequenced using HiFi. These tissue, oh, sorry, that's what I was just talking about. These tissue became a CiFi library, so by the time that Renee got this amount of tissue, it was like 1/6 of the fly. And if you can believe it, she still has some left. She didn't even use all of the tissue she needed, or she received. And then the rest of it became a Kinnex Iso-Seq library for annotations. So I had to modify my pipeline to now receive these 3 different data types. We've got the Fiber-seq, which is a HiFi that is used to create the contig assembly, that you combine with the CiFi to make it phased. Then that phase assembly gets scaffolded. Then you map the raw BAMs back to it to identify the regulatory elements. And then you use the Iso-Seq to do the gene annotation. And then you're done. So the data I'm going to be presenting today is on the melon fly, Zygodocus cucurbitae. So as the name suggests, it is a major pest of melons. So cucumbers, watermelons, cantaloupes, lots of different things you'd find in a salad. They attack like papaya and tomatoes too. So it's a big threat to specialty crops. Thankfully, it's not established in the mainland United States. It is in Hawaii. It's actually my favorite insect because it works for a lot of different things science-wise. If you guys are a fan of the Buffalo Bills, the quarterback, Josh Allen, his family actually has a cantaloupe and honeydew farm in California. So I like to say, if anyone wants to tell him, we're protecting his melons. But even if you're not a fan of the Bills, I think it's important, I think we can all agree that protecting American melons is really important. So one of the ways that we do this is through something called the sterile insect technique. If there was a large outbreak of melon fly in the United States, this is something that we could use to eradicate or suppress the population in 1 generation. So the way it works is you make a lot of sterile males, you release them amongst a wild population, and if they're lucky in love, then they'll find a lucky lady. But because the males are sterile, those ladies won't lay any viable eggs, thus eradicating or suppressing the population in 1 generation. This really only works if you can make a lot of sterile males. We can do that because we have something called the genetic sexing strain. So in a wild type population, everyone, males and females kind of look the same except for the ovipositor, right? So but the genetic sexing strain we have has a sexually dimorphic polymorphism that you can use to separate males from females before they're adults. So in a wild type, everyone has a brown pupae and everyone has like a standard chromosomal conformation or wild type chromosomal conformation. In the genetic sexing strain that we work on, there is a white pupae phenotype, so the females are homozygous for this recessive allele. They have a white pupae. The males are heterozygous. That's maintained by this reciprocal translocation between the autosome and the Y chromosome that fixes them as a heterozygous, so they're brown, females are white. You can separate them on a really large scale using an optical sorter. So the data I'm going to be presenting on is on this guy, so a heterozygous male that has a translocation. That's important. After doing the Fiber-seq HiFi assembly that's phased with the CiFi, we end up with 2 highly contiguous haplotypes. So I'm sure you guys have seen loads of these assemblies or this plot right now. It's called a snail plot. But the biggest take home here is that, so for this species, there are 7 chromosomes, 5 autosomes, 2 sex chromosomes, and both of the haplotypes are actually at a pretty much a chromosome scale as contigs. Usually when you phase an assembly, your hap1 and your hap2 end up way less contiguous than your primary. In this case, it's much less so because, as you can see, 90% of hap1 is in 10 pieces, 90% of hap2 is in 8 pieces. And of course, they're highly complete in terms of buscos. Okay, so this is the CiFi context for this hap1 and hap2. There are a few points I want to make about this. One, in hap1 and in hap2, you have these contigs that are at a chromosome scale, so that makes a lot of sense. The CiFi mapped really well and actually just really validates how good these contig assemblies are. We didn't need a lot of it, and we hardly needed any of it for assembly. And Kendall might hate me, but I had to do no manipulation in order to get them to look like this. I didn't move anything at all, or no like manual curation. I don't know if you could see, some of the contigs, or all of the contigs actually go straight through the centromere, which is pretty cool, because up until like long read data, the centromere is traditionally very difficult to sequence because it's so AT rich, and so things would often break. It's very difficult to sequence across and then assemble. But the long reads go right through it. So the centromeres, if that's what you're interested in, are intact. Let's see, what else did I want to say about these? Last thing is that HAP1 ended up with both sex chromosomes. And this is important because it was phased using CiFi and not trio parent-offspring data. So parent-offspring data would say like these haplotypes came from mom and these came from dad. But with CiFi, you're phasing where it says like this is one haplotype, this is another, but because the sex chromosomes don't have a buddy to go in the other assembly, they all ended up in one. So this actually means that the phasing went really well. So I told you this was a translocated assembly. That was a heterozygote for the translocation. So if you map those HiFi reads back to a wild type assembly, where you have in brown the autosome, and on the right side in gray represents the Y chromosome, you end up with some of the reads that are autosomal going all the way across the autosome, and some of the reads, half of them, map to the autosome and then continue mapping on the Y chromosome, which represents some reads having the translocation. So now that you have your really awesome assemblies, what we did was we annotated it using Iso-Seq only, using EGAPx. So EGAPx is an NCBI-generated program that to mirror their internal annotation program that they use to annotate genomes for RefSeq. So this is kind of the gold standard for when you want to annotate your genome, but they let us do it using our own data, but using their custom gene models, so it's going to be most highest parity with things that are in RefSeq. As of version 4.0 alpha, they actually let you use Iso-Seq as evidence, and as of version 5.0, you can use only Iso-Seq. You don't even need RNA-seq anymore. When you do a short read versus long read annotation on the same data set, what we end up is with the long read Iso-Seq annotated data set, you end up with, in our case, about 2% more reads, or 2% more annotated genes, 50% more mRNA isoforms. So this is, as Jonas said, like different isoforms from the same gene that weren't previously detected. We saw a 3-fold increase in non-coding RNA. So I think RNA is one of the, long non-coding RNA is one of those things that's pretty much invisible in short read data, but can be really important biologically because it can regulate genes both in distal and nearby and far away. Oh, and last thing is we saw fewer pseudo-transcripts, so fewer things that are spurious transcripts that aren't really real. Okay, so now we have annotated genes, really high quality phased contig assemblies. We can look at tissue-specific gene expression because we have Iso-Seq from different tissues, but also regulatory elements that are also tissue-specific. So these are just a couple examples. For ORCO, which is an odorant-binding protein receptor, what we see is, this is really hard to see because it's a really long gene, so the lines are very small, but we see expression in the head, no expression in the abdomen, so that's exactly what you would expect. And then accessibility in the head, which means that I am ready to be expressed in the head and probably not anywhere else. Its buddy, the odorant binding protein that it is a receptor for, also similarly has accessibility only in the head sample, not in the abdomen sample, which is good because there's no odorant binding in the belly, which makes a lot of sense. Okay, so finally, I just want to do one little snippet of our applications to a real study that we did. So I revisited an old study where we had insecticide resistance flies from the field that people, like growers, are saying, oh, they're not dying. So we brought them into the lab, compared them to our wild type colony, and showed that, yeah, they have lower mortality when you give them these to different doses of insecticides. And then we took those colonies and then selected for higher resistance. So we tried to kill them, we kept the ones that didn't die. Comparing pre-selected versus post-selected, you end up with much lower mortality in the post-selected flies. And then we did GWAS and Iso-Seq on these flies. And so this is just one chromosome, which alleles were statistically significant to contribute to the trait. And we saw that, one, in the different colonies that we got, like from different commodities, there are different genes that are under selection. So it indicates that insecticide resistance is a polygenic trait. Like, you can achieve it in lots of different ways. Sorry, I keep touching that. Two, what we see are some genes that are under selection, the alleles that are contributing or that are causing the changes in those genes, but also because of our better annotations, we have identified long non-coding RNAs that are possibly regulators of those genes. So we have gene, mutation, and what's regulating the gene. All right, so I'm way over time, but I hope I've convinced you that you can do a lot more with a very tiny amount of tissue. So there's only about like 10 to 15 milligrams of tissue from one of these flies. And from that, we got all these different library types, tissue-specific gene expression and regulation. We ended up with 2 really high-quality phased assemblies. I mentioned that one already. And using these better annotations, it actually creates a better foundation for the different applied research that we want to do on our species of interest. So with that, I'd like to thank the USDA for funding all of this. My dog, who missed out on a lot of walkies because I was doing data analysis nonstop for the last 2 weeks. This is our very, very small but very talented group of scientists that I have the pleasure of working with every day. And if anyone is interested in doing work similar to these, we have 3 postdoctoral positions open in our lab. Thank you.